Enzymopathies. Enzyme deficiencies causing hemolytic anemias. One is G6PD deficiency, glucose 6-phosphate dehydrogenase deficiency, pyruvate kinase deficiency, aldolase deficiency, hexokinase deficiency. So in the exam, if they give all the 4 options, all are correct only. So they will give the question as enzymopathies include G6PD deficiency, pyruvate kinase deficiency, aldolase deficiency, hexokinase deficiency, all the above. The answer is all the above. Glucose 6-phosphate dehydrogenase deficiency. This is a X-linked recessive disorder. Hereditary spherocytosis is autosomal dominant disorder, whereas glucose 6-phosphate dehydrogenase deficiency is X-linked recessive disorder, and it is characterized by hemolytic anemia on exposure to the oxidative stress. Pathogenesis. Normal red blood cells are protected from oxidant injury with the help of reduced glutathione. Causes for oxidant stress are viral and bacterial infections, certain drugs usage like antimalarials, primaquines, sulfonamides, nitrofurantoin, aspirin, vitamin K, and in conditions of metabolic acidosis and ingestion of fava beans, which is called as favism, will result in oxidant stress, thereby causing hemolytic anemia in G6PD deficiency. Favism can be asked for a 3-mark question or a 1-mark MCQ. Ingestion of fava beans will result in favism. Glucose is converted to glucose 6-phosphate with the help of hexokinase enzyme, and this glucose 6-phosphate is converted into 6-phosphogluconate with the help of conversion of NADP to NADPH in the presence of glutathione reductase. The glutathione is reduced, and the glucose 6-phosphate is converted to phosphoenol pyruvate with ADP generation with multiple enzymatic steps, and in the last step phosphoenol pyruvate is converted to pyruvate with the help of pyruvate kinase. This total cycle is called as Embden-Meyerhof glycolytic pathway, whereas glutathione reduction comes under hexose monophosphate or HMP shunt pathway. So the conversion of glucose 6-phosphate to 6-phosphogluconate. If there is G6PD deficiency, there will not be any reduction of the glutathione. What is going to happen in a normal person and in a G6PD deficient patient? Normal person, if there is a oxidant stress because of various causes, as there is glucose 6-phosphate dehydrogenase, there will be NADPH generation in HMP shunt pathway. As a result, the glutathione is reduced, and the reduced glutathione will protect the RBCs from this oxidant injury. As a result, there will be no hemolysis. But in the case of G6PD deficiency, glucose 6-phosphate dehydrogenase enzyme deficiency, when there is oxidant stress, NADPH is not generated in the HMP shunt pathway. As a result, glutathione is not reduced. As there is no reduced glutathione, RBCs are not protected from the oxidant injury. It will result in hemolysis. If this hemolysis occurs in the blood vessel, it is intravascular hemolysis, and if it is happening in the outside the extra blood vessel, then it is extravascular hemolysis. So in G6PD deficiency, both intravascular and extravascular hemolysis will occur. Types of G6PD deficiency. It can be type 1, type 2, type 3. Type 1 is mildest form, type 2 is moderate form, and type 3 is most severe form. Lab investigations for glucose 6-phosphate dehydrogenase deficiency. There will be intravascular and extravascular hemolysis. So the bilirubin, serum bilirubin unconjugated or indirect bilirubin will be raised. There will be raised urine urobilinogen, raised fecal stercobilinogen, serum globin, which is a globulin binding protein, will be reduced or absent in G6PD deficiency. The LDH, plasma lactic dehydrogenase enzyme, will be raised. There will be evidences of intravascular hemolysis in the form of hemoglobinemia, methemoglobinemia, hemoglobinuria, methemoglobinuria, hemosideruria. And in the bone marrow response, there will be increased reticulocyte count, that is, reticulocytosis will be seen. Hinge bodies are precipitated forms of denatured hemoglobin in RBC, and these hinge bodies can be revealed with the help of using supravital dyes or supravital staining. That is, the dyes will help you to see the cells in live form. So, hinge bodies are characteristically seen in G6PD deficiency. Bite cells are formed because of the splenic macrophages eating these RBCs to remove this hinge body. So, bite cells are characteristic feature of G6PD deficiency, which will result in intravascular and extravascular hemolysis.